Hi guys, Dr. Sweet's back to finish up my lecture on chapter 14, covering concept 14.3. So several times in the previous lecture, I mentioned how, Mendel was a little lucky or maybe things weren't quite as they seem. And that's what we're going to discuss in concept 14.3. Another one of those instances where I strongly encourage you to make sure you read through the book, get the terminology correct, and you might also find, as always, that you can Google around and find all kinds of help with all of these kinds of subjects, videos and things of that nature. They're a little harder for us to put into our lectures, but you're better at searching for that stuff probably than we are. So let's get started. I'm going to present a very short PowerPoint. I'm sure you'll like that part. Okay, so concept 14.3 is very succinct. Inheritance patterns are often more complex than predicted by simple Mendelian genetics. And the example down here, if you listen to my earlier lecture, should strike you because this looks like blending inheritance, the very thing that Darwin was trying to avoid. But I mentioned at the time that there are various situations in which you will combine two apparently alleles that seem to code for two opposite kinds of things, and it won't come out one or the other. It will come out blended, but there are reasons for that to happen. And to understand that, we have to look a little bit more deeply at what's really going on when we have an allele and what's that allele actually doing. So, dominance in a heterozygote turns out to really result in a gradient, excuse me. In other words, it may not always be strongly the supposedly dominant allele of the two. There may be all kinds of other things that can go on. We're going to go through that in this rather short lecture. So here's an example, much like what we saw before, where we have a P generation with a red and white flower. Now we have snapdragons. And these are true breeding, so we have homozygous individuals. And here we haven't used the terminology where we have the same letter, one very large and one large case, one small case, right? We have two different actual designations, the R and the W for the red and the white. And in fact, when we make our F1 generation, it turns out to be halfway in between. It's pink, a combination of the two. Now, what's interesting is when we go back and we do the F2 generation, what we find this time is that we still have not lost that white allele that's in there. And in fact, it'll come out again if we can return it to its homozygous state, which by the laws of probability, multiplication, and addition, we actually do. When we actually perform that and we have our genotype typical heterozygote cross of 1, 2, and 1, that actually turns out in this case to match the phenotype ratio, which in our previous example was 3 to 1, but now with red, pink, and white turns out to be 1, 2, and 1, where each genotype does have a different outward expression or phenotype. So, what is actually dominance? Well, dominance is just a way of talking about what the phenotype expression is of whatever the genotype may be. Now, remember, that can be not just some outward appearance like your earlobes and the knuckles on the hairs on your knuckles and eye color and stuff like that, but also physiological things, behavioral things, whatever can be affected by those genetics. How does that all work? Well, hopefully, as you've known from the past, when you were in high school and even junior high, and a little bit from what we've talked about so far, what really is going on for most genes is quite simple. The gene itself codes and tells the organism how to construct some kind of a biochemical, typically a protein of some kind. And often those proteins are going to be enzymes, but they may make all kinds of different things. So if the gene codes for a protein, and we look at something like the snapdragon here, what does the protein actually do? We have to ask ourselves. Well, in the previous example that we gave you with the peas, the purple flower, the purple allele beneath that, would actually produce a pigment, and that pigment would then suffuse into the tissues of the flower, and that would be a purple flower. The white actually produced no such pigment. And so there was no pigment going into the flower, and it was white. In that particular case, the purple allele was able to make enough of that pigment that it would suffuse the flower completely. In the case of the snapdragon, however, that red pigment is simply not made in abundance. And so in that particular case, think of it as being almost cut in half of what it would be. And so less red gets into the flower, and it turns out to be pink. Just a difference in how the operation and the appearance of that pigment actually works out in the two different flowers. So when we look at our previous case, we're going to have a term for that, and we're going to call that complete dominance, what we often think of as Mendelian dominance. And that means that the homozygous dominant is going to equal the heterozygote in its phenotype. In other words, the genotype of homozygous dominant and the genotype of heterozygous actually comes out appearing, or whatever it may be for the phenotype, comes out in the same state. But there are other examples. In the one we have here, this is an incomplete dominance, and that's going to be an intermediate expression in the phenotype, what we thought of as blending inheritance before. Remember that we have not lost whatever the original alleles were, because they are still particulate. They're simply expressed in the intermediate form, and that can happen for many different reasons, such as one that I just gave you. Now, when we think about the previous example, I talked about the snapdragon. The red pigment is producing a pigment, and the white is producing none. And so with half as much of the red pigment, you end up getting a pink flower. Now, it could be that the two alleles are both actually doing something, and both alleles may be expressed in the phenotype in some way or another. We call that codominance. In a way, we don't use that for our white example because it produced no enzyme, or no, in this case, sorry, no pigment whatsoever. But in codominance, both alleles are doing something and producing some kind of a biochemical, typically, and so both become expressed in the phenotype in some way. Probably one of our strongest examples and easiest to understand of codominance is our blood types, which we'll refer to probably many times in the next few years when we talk about genetics. You're probably familiar with the idea of ABO as your typical blood groups. And what are these various alleles doing? Well, these various alleles are producing, of course, some kind, in this case, of a carbohydrate. And that carbohydrate is attached on to the outside of your red blood cell. So when we talk about just expressing this as simply as we can with these nice little circles and triangles right here, obviously it's going to be more complex than that, but for our purposes, these symbols will do. So if you have this A allele, then the A allele will produce a certain shape of a carbohydrate that exists on the outside of your blood cells. And that's simplifying it a bit, but it works for this case. And the B, your B allele, produces a different shape of carbohydrate that's on the outside of those blood cells. So there's your A, and of course, if you were having both alleles A, then you would have that shape or that look to your red blood cells, and the B with those circles. Alleles are producing their various carbohydrates, and both are attached to the blood cell. So here we have an example of codominance. Both alleles are doing what they evolved to do, make a certain kind of carbohydrate, and we can see those results when we look, if that's what we're looking for, in their outward expression on the blood cell. So codominance, therefore, is a little bit different. And of course, what's nice about this is that the O group, this other allele right here, gives us an example really of what would be an incomplete dominance, because this would not produce anything. So this is closer to the snapdragon. One of the more telling examples of how complex this is, this will take me a minute to describe. It's described also in your text. You might want to read through this. There was a fairly good movie about this some years ago called Lorenzo's Oil, and I've kind of kept this on here for the last 20 years or so whenever I talk about this. But the road from genotype to phenotype when we start to examine this idea of complete dominance, incomplete dominance, and codominance, there may be various ways that that's expressed in that phenotype, as I've tried to describe all along. And so, in a way, our understanding of that, using this example of the disease Tay-Sachs disease, is that it depends on what level of phenotypic expression you might be interested in. And Tay-Sachs gives us an interesting example where, depending on what you're looking at, you can find either complete dominance, incomplete dominance, or codominance, again, depending on your sort of point of view. What expression are you actually interested in? So at the organismal level, we think of Tay-Sachs disease when we're really looking at the organism and whether a person is ill or not ill. And Tay-Sachs disease basically shuts down, in a way, your input and output. And so the folks that are unfortunately afflicted with this disease don't actually get a chance to communicate with the outside world, although they seem to actually know what's going on. At the organismal level, it's a complete dominance situation. In other words, homozygous recessives have the disease and heterozygotes do not. So that's complete dominance. Now, at the biochemical level, we might think of this as incomplete dominance, because here's what actually happens in this disease. The Tay-Sachs, if you don't have Tay-Sachs disease, a normal allele is going to actually break down a type of lipid, which is sort of a kind of fat, which we'll learn about more in your next semester, but actually can glom up some of the input-output systems with your nervous system. So in fact, if you are a homozygous dominant, you produce plenty of this enzyme that breaks down the lipids, and you are disease-free. If you are homozygous recessive, you produce none of it. These lipids, over a few years, usually it afflicts younger people, over a few years it will afflict that person as these lipids build up and cause these difficulties. Now, if we're looking at it at a heterozygous level, and we actually measure the amount of this enzyme that is produced that breaks down the lipid, well, actually it produces about half as much of this critical enzyme. So if you're actually looking at how much is being produced, because only one allele is doing the work, right? On your two chromosomes, one allele is doing the work, the other one's not actually producing anything. And of course, if you actually measure how much, a heterozygote will produce 50% as much of that enzyme as a homozygous dominant. But it turns out that that's enough. That will actually break down the lipid. And when that breaks down the lipid, the person that has that heterozygous condition turns out to be disease-free. So again, interested in that outward appearance, we think of as complete dominance. Heterozygote shows no sign of the disease. But if we look inside, we see that the actual enzyme was produced half as much, then that looks like incomplete dominance. It looks blended, halfway in between. Actually, if we think about that at the molecular level, and we really look closely, it turns out that our our recessive allele actually does produce something. It produces an object, it produces a chemical, but that chemical doesn't actually work. It's dysfunctional. There's been a mutation at some time in the past, and that one mutation, similar to what you've learned about with sickle cell disease and things of that nature, that one mutation means that it produces a useless enzyme that is still can be found in the body at that molecular level, but doesn't do anything. So when we think of the actual substances being produced, we can call that codominance. So here's three different kinds of expression that come out of the same disease. It all depends what level you're actually interested in when we use that term phenotype. It's a complicated example and can be confusing. You might want to talk to your professor about this one. And also go ahead and check in your text for some of the details. Oh, sorry. And if we haven't used this term yet, although I'm not sure if Dr. Stemke has or not, that's kind of mutation that we call often a loss of function mutation. It's a fairly common mutation, and it can be, quite serious. But that allele that's still producing this non-functioning enzyme has lost its function, even though it still produces something. We call that a loss of function mutation. Okay, so besides this codominance thing that I used with the blood groups, I wanted to point out something else that comes back here. And I'm not going to describe this because you'll come back to this at other times in your in your academic life if you've not already, which is how we have these, why you have to get your own blood type, universal donors and things like that with blood types, but I'm not going to go into that. I just wanted to point out that in this very famous example, we've been talking so far, as we did right at the start, very simplistically. We have these two alleles, purple or white, red or white. But obviously, when we think about the blood groups, guess what? There's actually three alleles. It's actually more complicated than that, but we're going to keep it simple for now. We have three alleles, the one that produces the A carbohydrate, the one that produces the B carbohydrate, and the one that produces really no carbohydrate on the outside of the blood cells. That's your ABO. So it's a three allele system, which causes its own kind of fun, which you'll find out probably when we get to genetics problems. It's a three allele system, and keep in mind that for most of our genetics, our genes that we have in any organism, there's not typically just two alleles. There are usually multiple alleles that one might have. Now, you only have two at that locus on the gene, correct? If there's only the one gene, at least. You only have two alleles in your system, and most organisms only have the two. But there are other ones circulating around, and depending on the next iteration of generations, there will be a different combination. You can be AA, you can be AO, you can be AB, you can be BB, you can be BO. I think I covered all of them right there. So all of those things could happen in a three allele system, and there are many more alleles than that for many, many different kinds of systems. So while we've given you these very simple cases, when we get to genetics, we should understand that there are multiple ways that these genetics actually work, and typically multiple alleles. That's especially true if you count these loss of function mutations, because when you look at a disease such as cystic fibrosis, which has many loss of function mutations that can cause the disease. When I say many, that means you could get this where the loss of function mutation happens at one part of the genetic sequence, perhaps in a different part of the genetic sequence. You can define those, if you want to, as different alleles, because one might affect base number 163 and one might affect base number 283. Those are different in a sense, even though they have the same result, which is a loss of function. So there may, in fact, be hundreds of different alleles for many different kinds of genes. Okay, what are some other variations that complicate our discussion of simple Mendelian genetics? Well, one is pleiotropy. So pleiotropy is the idea that a single gene, well, we've been talking in the most simplistic version, a single gene may have multiple phenotypic effects. And in fact, this is actually fairly commonly true, that the activation of a gene and the enzyme or carbohydrate or whatever it actually is coded to produce might have multiple functions within the system. One of the interesting examples of that is the SRY gene. Now, the SRY gene in humans is one that actually is on the Y chromosome. So males have the Y chromosome, as you know, XY. And when this SRY gene kicks in, it has this whole cascading effect where, because this one substance is produced, it ends up changing all kinds of phenotypic results, in essentially, to be honest, changing a female organism into a male organism, and all those effects that go along with that. So the SRY gene is one of the most common examples of that, but there are really most genes, or at least the substances they produce, may have many, many effects. Another example, which is a little bit similar to that, is what we call epistasis. And epistasis means that the gene, one gene may modify the expression of another. It's epistatic. The example in your book is a pretty good example. We have Labrador retrievers, which many of you probably owned. And basically, you have a couple of genes involved in the color of the fur of a Labrador retriever. One, for instance, might produce a pigment, while another gene and the biochemical it produces actually allows that pigment to be deposited into the hair. So the pigment's produced, but it's got to get into the hair in some way. This is actually true of a lot of mammals. Mice have the same situation. So if one produces the pigment and the other deposits in the hair, then you might find that a pigment's being produced, but if it can't get into the hair, then it doesn't really matter, right? So in this example from Labrador retrievers, we see our black labs, we see our golden retrievers, and we see our chocolate labs, the brown. And in this case, when we go through this sort of a system, you can see that our yellow labs actually don't get the pigment, into the actual hair. And so in this case, the gene, the alleles marked with the letter B talk about the type of pigment. It should be black or brown, and the E's are the one that actually introduced the pigment into the hair. So we notice down here in the bottom right of our dihybrid cross right here with the 16 possibilities, same thing as we've seen before right here. We see that we have four choices for our yellow labs. They're all, of course, homozygous recessive, so to speak, for the E allele, which means that they don't get the pigment into the hair. And so it doesn't matter whether it's black, whether we've got black or brown being produced, it doesn't get into the hair. So this is kind of interesting because notice that we have got a different phenotypic ratio that comes out of this because of this epistasis going on. Instead of 9-3-3-1, we have 9-3-4. But of course, if it wasn't for the way that this E gene operated, we would have different numbers of our black and brown labs. We'd have actually different numbers of the black and brown labs, and we'd be back to 9-3-3-1 in the end, and we'd only have the one down here, perhaps, that had no color at all. So, because of the way this particular system operates, we can see these different kinds of ratios, but they're usually variations on the 9-3-3-1. Okay, some other variations that come up, and this is also particularly common. And this is where you get into the more complicated situations in genetics. And you'll be talking about that when you get to genetics and in evolutionary biology later on. So we'll cover some of these things. Take a week probably to do it or more in later classes. But for now, we're just skipping over and just describing very briefly these different kinds of situations. Okay, so one of the more common of these is that there are a lot of kinds of phenotypic expressions that are not decided by just one gene, and in fact are what we call polygenic. There may be many different genes and alleles operating that control a certain kind of phenotypic expression. Probably the most one of the most common ones we can talk about in humans are skin color and height. And because we have a multiple of different kinds of different genes that are affecting, I think your book says, I think there are 160 different loci or genes that affect height. Then you can kind of add those together. If you think of them as being like units of something produced that might increase the height of someone, then your various combinations of homozygous dominants, heterozygous, homozygous recessives amongst all these different genes, and you sort of count them up as units, almost like money, then maybe you come out with different ranges of height and different amounts of melanin that are produced for skin color. That appears, of course, as always, to look like it's blending inheritance because you have all these different shades and colors and heights. And that's one of the reasons that blending inheritance was the sort of the paradigm of how genetics should work. Genetics, of course, back then they didn't know about genetics yet, but how inheritance should work back in the 1800s. And there was very good reasons, as I said before, for that to happen. But in actuality, it's still particulate. You're just actually adding those particles up, so to speak, to get the result in quantitative characters and polygenic inheritance. Again, that deserves a much longer conversation, but there's not time for that in this particular class. Just know that that happens because it's pretty important, actually, very, very common. And obviously, we have the nature-nurture debate. And the nature-nurture means that on top of all of the genetics that decide what some phenotypic expression might be, there may be many environmental factors that control that. 160 different, hope I haven't got that number wrong, 160 different genes that control height. But of course, if you're not getting proper nutrition, you may not be as tall as you could be. The genetics give you a possible range, you might say, but the environment may then decide how that's actually going to come out one way or the other. We see this also with soil nutrients and flower color. If you have hydrangea, like I do outside, my hydrangeas are usually white. but you can actually feed different kinds of soil into for hydrangeas and other flowers, and then it ends up coming out with different colors depending on what exactly uh, was the nutrient content of the soil. Things we might take for granted as being decided by genetics, like the red of a cardinal, northern cardinal, are actually decided by what they eat. So diet is a big part of the color. So these are what we call multifactorial. In other words, we have a couple of different factors, genetics and environment, that go into deciding what that phenotypic expression is going to be. But obviously, we start with these very simple conditions, but we should understand that all of these things can actually be in place for almost any one trait that you might pick out. It could be a combination of all of these quantitative characters combined with environmental factors. Some of those quantitative characters will rely on epistasis, some might be pleiotropic, so on and so forth. So know these different categories for this particular exam that's coming up, and we'll teach you more about that when you get to genetics and evolutionary biology. Okay, so I just want to finish this part of the lecture by saying that when we look back at Mendel, and I had this one lecture I just performed of what I call Mendel Extended, it acts as if Mendel was wrong in some way. No, he wasn't. His findings still provide the true basic underpinnings of genetics, and that is this idea of his laws of independent assortment and this idea of particulate inheritance. These underlie all of these various sorts of results in some way or another. It's interesting that Darwin and Mendel were obviously contemporaries. Darwin died in 1882, Mendel in 1884. As I said earlier, on the Origin of Species was published while Mendel was performing his experiments, and just a few years after it was published, he presented his papers, two important papers, and I think it was 1864 and 1865, to various in various conferences. After that time, the papers were actually published, and I believe the number is something like only 3, 4, 5 references to his work over the ensuing 35 years or so. So people didn't really understand what he was doing, and they kind of all thought it was all about botany. It was about how to grow plants, horticulture, and not so much about these more important underlying ideas. Literally, people didn't understand it because it was ahead of its time. It was way ahead of its time. As Mendel himself said to a friend in like 1869, he wrote and said, I can't remember the exact words, unfortunately, but something along the lines, I knew from the start that, this would be hard to understand because, it was ahead of its time. But eventually, he said, they'll understand. And in fact, as people started to repeat things like this, not because they read Mendel, but they started to think along these same lines, and they're still doing these experiments, watching inheritance traits, and they were starting to publish things that were sort of similar, and still, by the way, didn't really understand it. Then Mendel was rediscovered, and that was about 1900. And of course, both Darwin and Mendel were gone by that time. But in effect, when we think about Darwin and Mendel, we think about where biology and biological science is today with the modern synthesis, is melding of genetics, which Mendel had such a large part in actually pushing forward, even though it took a while for people to realize that, and of course Darwin and his theories of evolution by natural selection. So at that, I'm going to say that anybody who wants to can go ahead and cut off this lecture. And I'm going to go on and talk a little bit more about how to solve some of the problems that I talked about last time. So goodbye to whoever's not interested in that. It might be true. Your own professors might have their own way of explaining this. So you can watch it if you want, but you better do what your professor's telling you to do. We all sort of have our different ways of going about solving genetics problems. So goodbye to some of you. I'm going to go on for another 5 minutes or so. Okay, as you may or not recall at the end of my previous lecture, I was talking about how we could solve a trihybrid cross. And we can do that fairly simply if we simply know what the genetics are for each individual trait. And so I'm going to give an example here. And I have this, I think, at the end of my previous lecture as well. And maybe at the, I can't remember if I have it at the start or end of just the PowerPoint that I posted, and your professors will probably posted for you. So here we're going to look at flower position, stem length, and seed shape. And these were three characters that were studied by Mendel. Now, to present you with some questions to do with the dihybrid cross. Now, what I'm going to do is I'm going to take just a minute here and talk a little bit about how we can sort of understand this. So I'm going to change what's on my screen here. I think this will work. If not, I'll have to cut it out. And what I want you to sort of think about for a second is to think very simply about the situation where we're doing some kind of a cross and we're concerned with only one trait. What are the various possibilities of that cross? So we're talking about a system in which we're talking about a single gene that has two alleles. So we're not talking about some situations like in plants that we'll get into later. So most animals, for instance. And what are the possibilities? What kind of crosses can we think of? Well, here we have a very simple one, right? I've already typed this out. This is obviously a homozygous dominant crossed with a homozygous dominant, right? So let me move that over. Homozygous dominant and a homozygous dominant. Okay, that's pretty simple. We don't have to think about that very much because, after all, we're just talking about 100% one phenotype and 100% one genotype. It's going to come out the same no matter how we cross it. About the same situation is true if we do that in crossing a, sorry, screwed that up. If we cross a, still I'm doing something wrong, sorry. That's what it is. If we cross a homozygous recessive with a homozygous recessive. One there. That's obviously very simple and almost really, in a way, too simple to even talk about, I guess I would say, because it's beyond, we don't have to really think about that very much. But you cross a homozygous recessive with a homozygous recessive, the genotype all comes out 100% recessive, and the phenotype comes out 100% recessive. And of course, we've already talked about our heterozygous cross, right? So here's our heterozygous cross. And in our heterozygous cross, we've seen this already. Here we have the two alleles produced by the law of independent assortment out of either the male or the female. It really doesn't matter which. And we know what this result comes out to be, right? It comes out to be genetically a homozygous dominant, two heterozygotes, and a homozygous recessive, a 1-to-1 ratio, 25%, 50%, 25%. Phenotypically, in the case of complete dominance, which is all we're talking about here, we have three that are come out with a homozygous, with the dominant trait, I should say, excuse me, with a dominant trait, and one that comes out with the recessive trait. So the phenotypic ratio is 3-to-1. Well, let's look at some we haven't talked about yet. So I'm going to go ahead and type some of these in right now. So we could cross, for instance, a homozygous dominant with a homozygous recessive. Let me go ahead and let me correct this spelling error here. So now we're going to cross a homozygous recessive with a homozygous dominant. How does that come out? Well, this is pretty simple as well, isn't it? We have R, right? We have R, little r, little r, R, R. In every case, because we just fill in from the row and the column. That's how Punnett square works. And now we see that we have a situation where the genotype is, of course, 100% recessive, homozygous recessive. And the phenotype is, of course, God, I screwed that up, didn't I? See, I'm thinking about something else I'm doing. Sorry, the genotype is 100% heterozygous. I'm already thinking about the next thing I'm going to do. Jeez. And the phenotype, therefore, is going to all be in complete dominance, is going to show 100% the dominant trait. Okay. It's one kind of cross. I got to shrink these down. Because I got more to do. All right, so now I'm going to go ahead and go to our other examples that we have. Let me draw a nice thick line underneath this so we don't get these all messed up. Thick bottom border. All right. So here we're going to cross something a little bit different. Now we're going to take a heterozygote. We're going to cross it with homozygous dominant. Okay, so we're going to do a heterozygote with a homozygous dominant. How's that going to come out? R, little r, r, little r. Okay, that's another kind of cross. What's this genotype and phenotype? I'm just going to copy these over here so I don't have to repeat myself all the time. Here we see the genotype is going to be a 1-to-1 ratio, isn't it? The genotype is going to be 50% heterozygous, and it's going to be 50% homozygous dominant. Make that smaller. It's no good either. I have an idea. Let's see if I can fix this. So I give for doing this on the fly. Oh, they can't do that. Okay, we'll make it a little smaller. So hetero, then homozygous dominant. Okay, so what about the phenotype? Well, the phenotype under complete dominance is still 100% dominant. So that's good. So much easier if I was on the blackboard. All right. So what's our last possibility? Hopefully you've thought of this already. We can cross a heterozygote with homozygous recessive. Now we've crossed. Do it this way. Crossed a homozygous recessive with a heterozygous individual. All right. Now how does that come out? That comes out big R, little r, big R, little r, and 2 little r's. What is that going to look like? That means that the genotype is going to be 50% heterozygous, 50% homozygous recessive, a 1-to-1 ratio. And here I'm just going to copy this just to be a little easier. Phenotype is going to be 50% dominant and 50% recessive trait. Okay, so why did I go through all that? Well, I did that for a couple of reasons. Hopefully I haven't lost you yet. Sorry. I want to shrink all the, I know, let's do this. Hang on. There you go. So you can see the whole thing. Those are your 6 different crosses of a single trait. And if you can spend the time to sort of think through these 6 crosses, you're going to find that you're going to be able to answer a lot of the questions a little bit more easily. You might actually just write these down and have them on a sheet next to you when you're doing your genetics problems. And so if you do that, then you can kind of quickly look over, remember what your ratios are going to be. Remember, you always got, when you're doing genetics problems, you always got to think about what's being asked. Are we being asked for the genotype, or am I being asked for the phenotype? A lot of students get messed up with that. Well, using this little little thing I have right here, my little Excel spreadsheet, let's see if we can figure out what that rather complicated example was. Try to show 2 things at once here. I don't know if this is going to work, but it's always fun to try these kinds of things. Try to keep them both up. There we go. So my first question was, if a plant was heterozygous for all 3 characters and was allowed to self-fertilize, what proportion of the offspring would be as follows? Well, the first is going to say homozygous dominant for the 3 traits. So what proportion are going to be homozygous dominant for the 3 traits? Well, we're looking at a heterozygous cross. Let's go over to the heterozygous right here, right? And if we treat each one of these, one will be round and wrinkled, one tall dwarf, one axial and terminal. And so if the proportion of offspring and homozygous dominant for all three traits, we can see that we have a situation where only 1/4 of the offspring for any one of the traits is going to be homozygous dominant. So now we can multiply our three traits together. 1/4 times 1/4 times 1/4. And 1/4 times 1/4 times 1/4 equals 1/64. Probably should pause now and let you guys try this out. But, homozygous recessive for the three traits. If you're looking real closely, you can see that I've actually allowed you to see the various the answers down at the bottom down here. Homozygous recessive, you can do the same way. What's a homozygous cross for each of the three? The homozygous recessive for any one is going to be 1/4. So again, I have three traits. 1/4 times 1/4 times 1/4. That's 1/64, because 4 times 4 times 4 is 64. How about heterozygous for all three traits? So far, I'm asking all genotype questions, right? Well, obviously, 1/2 of any one of the three traits is going to come out as a heterozygote. Well, I've got three traits, heterozygous for each one. So 1/2 times 1/2 times 1/2. 1/2 times 1/2 times 1/2. 2 times 2 times 2. That's going to be 1/8. Okay. Heterozygous dominant for axial. Phenotype dwarf. Heterozygous for seed shape. Ooh, now we're getting crazy, aren't we? All right. I'm going to think about this one a little bit. Homozygous dominant for axial. Okay, well, doesn't matter. I'm still crossing heterozygote. So homozygous dominant, we know, is 1/4, right? The phenotype is going to be dwarf. I got to check and see what my dwarf number is. It's recessive. Okay, so 1/4 for the first one. I'm going to do it again, and the one that has a recessive phenotype, also 1/4. All right. Heterozygous for seed shape, but we know heterozygous is 1/2. So it's 1/4 times 1/4 times 1/2. 1/4 for the homozygous dominant, 1/4 for the recessive phenotype, and 1/2 for the heterozygous. 4 times 4 times 2, 32, 1/32. Flowers axial, stems dwarfed, seeds round. I can do the same thing. All I got to do is look at one cross. Don't get confused. These are like variables in here, right? Flowers axial. Okay, that's asking for a phenotypical trait, phenotype. The axial is dominant, so that's going to be 3/4. Ooh, this can be fun, isn't it? Stems dwarfed, we know that already. That's going to be 1/4. And seeds round, and that is a dominant trait. So the dominant trait is 3/4 times the recessive 1/4 times the dominant 3/4. 3/4 times 1/4 times 3/4. 4 times 4 times 4 is 64. That's on the bottom. 3 times 1 times 3. That's in the numerator. That's 9. The answer is 9/64. So instead of getting hung up in a tri-hybrid cross, which we never gave you, that would be like taking the original our original cross that was, I'm not sure I have one on this spreadsheet, sorry, but our original dihybrid cross, remember, had 16 squares in it. Well, if you're going to do a tri-hybrid, it ends up being 4 of those together. And so that ends up being 64 different slots that things can be in. And it seems very confusing. But if you simply follow the simple rules for a single cross, and it could be any one of these. I was doing all heterozygous crosses, but it could be any one of the 6 that you see right here. Any one of those 6 possibilities. You follow those rules, multiply them together for the different traits, and you'll come out with the right answer. Okay, well, that was fun, at least for some of you, and hopefully you'll enjoy that. And I know that we talk about it in my class as we go on. I'll see you again in a few weeks. Let's see, when will I see you again? Dr. Mills says I'm going to see you again on, for Lab 4. That'll be fun. I guess on the 15th of September. Okay, see you then. Have fun.